work, and we're still going at it. But she got a position at UCLA in this amazing lab that harnesses human cortical spikes and subcortical spikes. And so she's going to be applying all these methods to analyze that kind of data. So it's really exciting for us. So this is a paper she published this year. The autonomic nervous system differentiates between levels of motor intent and end effector. And so I'll discuss the paper because it's relevant to the whole discussion of autonomy and detecting this kind of stuff. This was a graphical abstract for the paper. It made it to the cover of the Journal of Personalized Medicine, and it's doing quite well in the journal. So there's a lot of interest out there as it turns out. So the first thing is to note the sort of taxonomy of the nervous system that we are proposing with the different levels of functionality: autonomic, involuntary, spontaneous, and voluntary. And in this particular paper, we combine autonomic and voluntary versus spontaneous type of activity in an experimental essay that will become clear in a moment. But the idea was to combine central nervous system, that is the brain and the spinal cord, with information about the peripheral nervous system, including the heart. And so in this paper, we focused on peripheral nervous system through kinematic parameters and heart activity, and made inferences about the central nervous system, which controls intent and deliberateness in our actions. Now, again, we need to make this distinction between mental intent and physical volition. So again, that's sort of the ongoing theme, and bringing methodology to the table that enables us to utilize the peripheral nervous system as a proxy of what the central nervous system is thinking or doing, and with the corresponding caveats, of course. So then the other thing that we did in this study was we represented the entire body as a network, interconnected network, with nodes that we represented with different metrics, cross-correlation and cross-coherence and clustering coefficients within the network and things like that. So we built representations of these nodes as an interconnected dynamically changing network, and study it under the deliberate and the spontaneous level of activity, combining the kinematics with a heart rate signal, and then making inferences about what most likely was happening during the action. And what we found was a segregation of activity that was deliberate in some parameter space versus activity that was spontaneously happening, and also activity that was performed by the dominant, by the performing part of the body versus the non-performing part of the body. So we were tracking simultaneously 10 parts of the body in the upper body as they were performing a decision-making experiment, and also involving some memory component and so forth. And so we could tell you the part of the body that was actually performing the action versus the other part that was kind of passively there. This is the experimental assay that G developed in collaboration with the rest of the lab, actually, because there was a lot of feedback in doing this. So she presents as a touch screen, and she presents a signal to touch. And so she collects the responses, the touches and so forth. And after the touch, she plays a high tone, which is variable in size. It could be 100, 400, 700. And then you have to point and touch where in this scale the tone is, whether it is in the how much time it elapsed, whether it was in this region or in this region and so forth. And we're collecting all the information about it. And this takes about 10 seconds, the whole trial. So these are the epochs. So the person sits there and it touches. So we collect the touches continuously, all the kinematics and the heart activity. They're wearing sensors and they're also wearing a heart monitor. And then they wait and listen for the tone, and then they actually point. So we're collecting the whole unfolding activity at the periphery. These are the locations of the sensors that we're going to focus on. We had more sensors, but we restricted it to the upper body. So this system has like 15 sensors, but we restricted it to 10 in the upper body and the head. And it collects data at 240 hertz, and it's integrated with an interface. This is a touch screen in an iPad, and so this is all integrated with within an interface called the motion monitor that makes sure that everything is synchronized and nicely collected and so forth. Any questions so far? Okay, so that's just the instrumentation that we use. And then we broke down the analysis based on these levels of forward, deliberate, and backward uninstructed, which is more of a spontaneous retraction of the hand, but also on dominant versus non-dominant. And by dominant, we don't mean like right versus left dominance, but we mean performing part of the body, like the performance side of the body. And so they give information about one thing that we were interested in was understanding external goals and the coordination of movement and response of external goals, which are visual and auditory, and also in internal space of coordination in the joints and the body, the executing body in motion. And so that was sort of like a breakdown of how to organize our thoughts about it.
What is dominant and dominant again?
It's the dominant is the side of the body that is performing the action, like your right hand side, okay. But it's not like, it's not your dominance. Like you could be left-handed, but be performing with the right side of the body. So it's just the side of the body, independent of the dominance that you have.
Okay.
And then we analyzed the kinematics network and the kinematics heart network. So both, so that means like including only the kinematics of the body, but also adding the kinematics of the heart signal, both for forward and backward and for dominant and non-dominant. And then, so that's just a kind of a breakdown of the organization of like what we're doing with the data that we're collecting, how we're integrating it and so forth. Then we have, now we take the data and we derive this is biophysical data. So we derive the micro movement spikes, which we have explained in the past. And then we derive an adjacency matrix. An adjacency matrix in each entry, if you have a value of 0, means there is no connection between those nodes. So you can think of it as a pairwise representation of different nodes. So if we have 10 nodes in the network, and the entry 1, 2 is the link. If there is a link between node 1 and node 2, there is a value there. Otherwise, there is a 0. That's just, it's called an adjacency matrix. And we use different representations. Usually you can use similarity metrics of different kinds. And we, in this particular work, we use cross correlation, which is, it's a linear type of methodology, but it's good enough to represent a similarity in terms of synchronicity and lags of the synchronous periodic activity between two body parts. So we look at that pairwise for each one of the 10 body parts, which each one of the other remaining 9 body parts. And that gives us a matrix. So we'll see that in the next figure. But with the adjacency matrix, we can convert it, represent it as a interconnected graph weighted directed or weighted undirected, and do connectivity analysis and come up with like equivalence of synergistic activity that forms self-emerging clusters of activity, self-emerging subnetworks, and we can track a number of things in the network. And so G focus on clustering coefficient, which gives you a sense of a clustering network activity that is happening simultaneously, and given a node with the surrounding nodes. She looked at the median clustering coefficient across the network, and so she also did analysis in the spatial domain and in the temporal domain. So yes.
So one question regarding the heart sensor. Is that the metric is heartbeats?
Yeah, so she used interbit spike timings.
Oh, okay, I get it. Yeah.
Yeah, so we can use amplitude, and I have to see, I think she might have used the amplitude of the signal as well, the fluctuations, but I think she focused on the interbit timings. But we can use amplitude of the signal and fluctuations in the amplitude of the R peaks. So it's like a complex of maybe Mona can tell us.
So that's like ECG data. That's almost ECG data.
Yeah.
Okay.
Yeah, I think yeah, we usually do like the IBI, and then you can also do the full ECG signal, which I think this is what G focus for this.
Yeah, I think G took everything because, and this is what this slide speaks to that. One of the challenges is to not induce fatigue, so that because we want enough peaks in the signal to make a good tight confidence interval estimation, we have several signals at our disposal. So there's a kind of a trade-off here. One is like, do you get fewer peaks but more accuracy, less error, because first order approximation. So you have position, right? And then you get velocity, and from there you get speed. So that's the first order parameter. So your error is lower in differentiating from each one of the points in that trajectory, because there could be sampling error. And if you differentiate it, you will amplify that sampling error. And so that's a trade-off. If you look for higher order derivatives like acceleration or jerk and so forth, that gives you, so you have to actually be very careful with this trade-off. But that's what we did. We have filters, we have signal processing methods that allow you to explore different versions. So we looked at linear speed, we looked at angular speed, we looked at linear acceleration, and we looked at angular acceleration. And we could get the most number of peaks in the angular acceleration, obviously, in the shorter amount of time, so we don't induce fatigue. So we can kind of reduce the number of trials to get enough confidence intervals under the sample resolution that we have. Any questions? And so but we use all of the signals. So the linear speed is good in telling us the pauses of the system when it's going forward and then automatically retracting backwards. I mean, I should say spontaneously, because there's a difference between automatic motions and spontaneous motions. And the difference, as we will discuss later in the microfacial expressions, is that spontaneous motions is something that you can bring up to consciousness, you can bring up to awareness and turn into an intended motion. But if you bring up to awareness an automatic motion like bicycle riding, it will interfere. It will interfere to try to control that. So that's the fundamental difference between a spontaneous action and an automatic action. So I want to be careful, because the uninstructed retractions of the hand, which happen without even the awareness of the person, are spontaneous in nature in that sense that if you bring that awareness, then it becomes intentional and you can train it. Then you can retrain. You can train it purposely. But you can also train it like sort of through the back door without telling the person, and you're training the forward movement, but in reality, you're adapting actually the spontaneous retraction. So there are all kinds of tricks that you can do here. And again, this is a model that speaks back to the Skinnerian, approach to behavior modification, because exactly what's happening with this social media thing. You can think of the spontaneous retraction as the part that is beneath awareness, and you don't know you've been fed certain information that is modifying your behavior unconsciously. But you can bring that up to awareness and then kind of mitigate the effects of the consequences of training your system through reward-based training. So the second part of this is that we then look at the distributions that we get from the micro movements once we settle on the parameter identification for analysis and to optimize this fatigue thing. And so then after that, we run our maximum likelihood estimation algorithm and begin to see what distributions are most likely the ones that will work for our data. So we settle on the gamma because even though we have other competing normal and non-normal for the micro movements, we know that the gamma is more general, and we'll see, this is like one case, but we'll see cases where you get exponentially distributed data that when you pass it through the micro movement reparametrization will not be nicely like this, will be more skewed. So we'll have heavy tails. So that's why we settle on the gamma. This is one example of it. You have a heavy tail here. And so then the first pipe of analytical pipeline, again, takes the position trajectories forward to the target of your decision and then retraction, takes the angular acceleration, derives the micro movements to examine the amplitude of the fluctuations in the amplitude of the signal, derive the micro movements, and then look at two different body parts to construct pairwise our measures, and then look at the gamma characterization of different body parts and at different times. And so this is kind of the pipeline of that we've already explained for other data analysis. What then becomes new here is that we take cross-correlation of linear speed, and in this case, it's a cleaner signal to get this cross-correlation, and then we build this, and we don't need as many peaks from it to build it. It's just a simple cross-correlation matrix. We build our adjacency matrix where you have every entry here represents a link from this node to the other node. So this is like entry 1, 2, 3, 4. So this is 1,4. And so we go to link 1, and that gives you the amount of maximal cross-correlation between link 1 and link 4. Maybe link 1 represents the head and link 4 represents the hand, the right hand, for example. And then we study connectivity metrics from this network and have a network representation and study things like the clustering coefficient and the behavior of the neighbors to a node. And then we represent this graphically by having, for example, the size of the node representing the maximal cross-correlation, and the length and the thickness of the length representing, for example, the lag. So you can see which body parts are maximally cross-correlated with other body parts, and what parts are leading, and so forth. And you can have other representations. You can use other parameters to visualize your network. And so this is the second pipeline. Now is when we combine the IBI activity. Actually, G is collecting not just the R peaks, but also the other peaks. So as Mona was telling us, she's looking at the at the entire signal fluctuations of the amplitude of the entire signal, not just the inter-peak interval timings. So we get the angular acceleration, and we get the ECG data and convert that to micro movements as well. And then we represent their histograms and fit the probability distributions. And we do cross-correlation analysis of that signal, of the heart signal with the body signal. And then we study the network representation again, utilizing, for example, things like the earth mover's distance, which I don't think she explained here, but essentially, as we explained in another class, gives you the amount of work that takes to transform this histogram into this histogram. So it's kind of a metric of effort, but we interpret as a metric of distance in probability space, given the framework that we're using. And in this case, she's representing the air mover's distance pairwise taken across the different limbs. So this is the head, the shoulder, the upper arm, the forearm, and the hand. And the color represents which signal is leading, whether it's the heart signal or the kinematic signal in blue. And the thickness represents the amount of that is leading ahead. The signal is leading ahead. So it enables us to visualize immediately which body parts are leading, which signal, whether the autonomic signal or the kinematic signal is leading or lagging. Any questions so far? And so the idea is that once we have these representations and we look at a random draw of the population, we can see what signals are automatically informing us of the level of intent that the system has, based on, because we know our ground truth, we know what is intended and what is spontaneous. So now it's like, once we characterize those signatures, we can flip the question, and given that signal, what most likely is the state of the mind that the person has. So in this sense, we're using these biophysical signals as a proxy of what the brain most likely was thinking at the time, or what state of intentionality the brain was. So then we can differentiate different levels of intent that way. So this is using the noise to signal ratio in the gamma probability distribution characterization, and the difference. So this is the angular acceleration, and the difference in the noise to signal ratio between conditions. Then we can look at the forward, at the signatures due to the forward kinematics, and at the signatures due to the backward kinematics for each participant. Each point represents a participant, and it automatically begins to show this division, except for one subject which fell in the backwards, the forward fell in the backwards. And you expect these kind of variations. It's not a perfect, but as you add more participants, like if we had 100 participants, we would see the clustering emerging quite nicely. And this is the same kind of analysis for the noise to signal ratio between the dominant and the non-dominant. Here we see a farther apart cluster self-emerging from all the dominant hands had a higher difference in noise to signal ratio. And so, yes.
So I had one question. How do we denote this disambiguation to agency and not the fact that one is forward and other is backward?
I'm sorry, what, how?
So you were saying that forward and the backward seem to split up quite nicely, but why is that why are we denoting that separation to agency rather than to the fact that one is forward and the other is backward?
No, it's not.
So there is bound to be different.
It's not agency directly. It's the contribution that this makes to agency. So the forward one, we know is intended and goal directed.
Yeah.
The backwards one, we know is it's not instructed and is beneath awareness.
Yes, but what I'm saying is that the difference might simply be because one is forward and the other is backward. And so for me, the strength would be more if both were forward, but one was intentional, the other was not intentional. See what I mean? Like the
Yeah.
movements themselves are different, so the signatures would be different. And
yeah, but so you're right in that the movement directionality is different. The directionality of the movement is different, but the kinematics, if you go back to the trajectories, are indistinguishable.
I see. Okay.
So, but you're right. I mean, a better assay is to have the person do something spontaneous in the same kinematic direction as it did it deliberately.
Yeah.
And then,
for example, I'm thinking of an experiment where one is a pointing, so the backward movement is obviously spontaneous, but the other one could be you point, but also you bring something back, like maybe a tiny small object.
you can design.
That would be an intentional backward.
Yeah, we designed an experiment like this where we had multiple motions in which you had the same biomechanics, but in some cases you had the exact same biomechanics, but in some cases you're traveling along a deliberate path, and in other cases that is just a transitional path. We have that data as well. Okay. But in this experiment, it was simpler, just forward and backwards. So the spontaneity of it is just pertinent to the action itself. It's not the same exact direction. Is everybody clear on this difference? It's very subtle, but it's very important, right? Because it makes a stronger point. But we'll see that one paradigm that will lend itself so nicely to do this is the face, this facial microexpression. So we'll be able to stage the expression and get the expression spontaneously. Like Richard did this with, and Mona also, they did this like staging a particular expression and then enacting it. And in staging it, actually, you can transition to the actual expression. I'll show you at the end of the talk a situation that Mona G and I got ourselves into, and now I'm studying it to see how we can precisely address that. But here, what we learned from this was when we represented the network, we realized immediately the dramatic differences in doing the forward and the backwards motions in the network representation itself. And here we had an asymmetry in the body. And we actually did this first, and then that prompted us to look at the data in these terms. And then we saw that the asymmetry was even stronger, segregating. So that was quite interesting. And also the connectivity then alerted us that there was something going on with the lag information that we could further explore. And that's when we decided to incorporate the data that we had analyzed. We had the data from the heart, which we had analyzed separately, and then we decided it's really important that we do these, that we integrate both data sets. And so here is an analysis of the clustering coefficient of the network connectivity of the kinematics only, and using the median cross correlation differentiate between the levels of intent. So forward is a truly intentional motion and goal directed. It's unambiguously intentional. Backwards has a lower level of intent because you weren't really instructed to do it, and you didn't really have a goal other than go back to rest. So this level of intent is lower than this level of intent. And so we can differentiate it using this network representation. The the size of the circle is the median clustering coefficient of the network, and the thickness of the link represents the lags of the cross correlation that we computed. And so you could see differentiation in different body parts, and also the asymmetry that keeps propping up with a dominant versus non-dominant hand. So that was also using the cross correlation median of the linear velocity. The clustering coefficient median of the linear velocity versus the maximal cross correlation, and that also differentiated the two. And then that's when we realized that incorporating the heart signal would be very informative, and indeed it was. And so we did the maximal cross correlation, the lag, the corresponding lag, and the air mover's distance, measuring the differentiation between the gamma estimation from the micro movements derived from the signal itself. And so the network connectivity analysis here was incorporating the heart signal and the kinematic signal. So we could tell then which signal was leading which. And here we're representing 1 over the air mover's distance. That's the size of the circle, and the thickness represents the leading information, and the color represent the prevalent signal. So in this case, the heart was the prevalent signal, was the leading signal, and it had a clear engagement of the part performing, of the body. This is the right side of the body performing the activity, whereas the spontaneous retraction backwards had a prevalence of the kinematics. So when the kinematics was intended towards the target, the heart was leading the kinematics. When the task was the spontaneous retraction, the kinematics was leading, and the lead happened precisely in the hand and the forearm performing the task. This is all automatic, so we don't. This is all from the variations on the fluctuations in the signal. We're not doing any kind of machine learning training and testing thing here. It's all from the signal itself, and we could see that then in a parameter space, and also build that parameter space for the dominant versus non-dominant hand, and look at the signal itself, and then at the differentiation of the signal. So that's also informative. So this was the result. So that given now the kinematic signal and the ECG signal itself, characterizing it in these parameter spaces could tell us which one was the dominant side versus the non-dominant side, and which one was the forward versus the backwards movement. So it serves to, given continuous signals, characterize, infer back most likely the state of the system, whether it's the dominant side, whether it's the intentional part of the task, or whether it's the unintentional part of the task. So that was a paper that G published this year, and the summary is that we can use biorhythmic time series signal to detect levels of intent and characterize it so that we can infer back. We can detect lead lag dynamics across different levels of neuromotor control and infer, given the signal, which mode the system is most likely in. In the deliberate mode, we know that the heart leads the kinematics, versus in the spontaneous mode, the heart lags the kinematic. And we also can detect the body part that is actually performing the action. And so in future work, we want to do this in autism and in other disorders of the nervous system. So questions? Any comments or questions? So it's 3:30. Why don't we take a break until like 4:45 or so, and then we delve into the 7 universal micro expressions, which is a lot of fun.